we go. Okay. So smell and taste are related in the sense that our brain sort of combines them to get an idea of what flavor is. Okay. The cortex that processes taste is primarily in the insula, which is that fifth lobe of the brain that's tucked between the parietal and temporal lobes, sort of on the surface, the lateral surface of the thalamus. And the cortex for smell is in the temporal lobe, okay? But what happens is that as we get sensation for taste and smell, those get combined before they get sent to the frontal lobe, and we decide what we want to do about what we're experiencing, if anything, okay? Both of these systems use chemoreceptors. The taste buds are, of course, on the surface of the papillae of the tongue, and the olfactory epithelium is in the top of the nasal cavity, okay? And these are both chemoreceptors, which means they are ligand-gated channels, meaning a chemical binds to them, a channel opens up, and ions move through that channel, okay? And that results in a depolarization that results in what we call a receptor potential, which can result in neurotransmitter release. And then the transduction happens with the afferent neuron, right? So the olfactory epithelium in the roof of the nasal cavity is going to cover the superior nasal conchae, which is we'll see in a minute, a ridge on the lateral margin of the nasal cavity. It is part of the ethmoid bone, okay? And its purpose, the nasal conchae, is to increase the surface area available inside the nasal cavity so that you can filter, humidify, and warm up the air more effectively to prepare it to move down into the lungs when you breathe, okay? So it doesn't damage the lungs. The olfactory sensory neurons are actually bipolar neurons with olfactory cilia surrounded by supporting cells that cushion them and stem cells at the base of the epithelium. So this is, an unusual, this is cranial nerve 1, okay? This is cranial nerve 1, okay? It's the shortest cranial nerve, okay? It goes through the cribriform plate of the ethmoid bone. Okay. And connects the nasal cavity to the olfactory bulbs. Okay. It is the only cranial nerve that regenerates. Okay. And part of that comes from these stem cells, okay, which are within the epithelial layer, right? But part of it also comes from something called the rostral migratory stream, which are, they are neurons that migrate to the olfactory bulb, differentiate, and then function. Okay. And it turns out that the signals that probably do that are lying in the glial cells of the bulb that call them to that location. Okay. And I think we talked in lab about the fact that this trick, this unique property of these olfactory neurons has been used to actually fix spinal cord injuries successfully, at least in one case. Okay. So this is an unusual sense. Okay. So you can see here what happens is that you inhale odorants that dissolve in the mucus of the olfactory epithelium. The bipolar cells are going to carry that to the olfactory bulb. Okay. And in the olfactory bulb, so we can write a pathway actually. So cranial nerve 1, right, to mitral cells in the olfactory bulb. Okay. And then from there, okay, there are olfactory tracts that project to the temporal lobe. Okay, that's the pathway. So this is the only special sense that bypasses the thalamus. Okay. You can see that there's 3 nasal conchae, right? There's the superior, there's the middle, and the inferior, right? The inferior is its own bone. The superior and middle are part of the ethmoid bone. Okay. And there's the cribriform plate right here. Okay. And it has holes in it so that those neurons can stick through and mediate our sense of smell. And the reason that these neurons can regenerate is because they're exposed to the outside world. And that means they get beat up, and that means they need to be replaced. Okay. So the apical dendrites terminate in the knob. Large olfactory cilia radiate from it, covered by mucus, which is the solvent for the odorants. Non-myelinated axons of the olfactory receptor cells gather in fascicles and make up the filaments of the olfactory nerve, cranial nerve 1, and they live from 1 to 2 months. Okay. And then they get replaced. So you can see here, right? Those are the dendrites, right? These are the neurons. These are bipolar neurons. Bipolar. Okay. And then you've got those supporting cells that help them out. All right. And then you've got these stem cells. All right. And then you've also got these cells that come from the rostral migratory stream that walk up to there and do their work. Right. There's the mitral cells. Right. And these are glomeruli. All right. This is where we synapse the bipolars with the mitrals, and then we project back, and that's going to ultimately go to the temporal lobe. Okay. Smells have hundreds of different odorants. Humans have about 400 smell genes in the nose, each encoding a unique receptor protein, which responds to 1 or more odors. Each odor binds different receptors, which have 1 type of receptor protein. There are also pain and temperature receptors in the nasal cavities that respond to nasty stuff like ammonia or extremely hot or cold chemical odorants, right, that are out of the range of the olfactory receptors. Okay. The odorant has to be gaseous in order for it to be smelled. It also has to be able to dissolve in the mucus. Okay. The odorant binds receptors on the membrane that open anion or cation channels, and then in usually goes with sodium. Okay. And that threshold, the action potential goes to that mitral cell. Okay. In the synapse at the glomeruli in the olfactory bulb. Right. Odorant binds the receptor as a G protein. Okay. Which causes cyclic A&P synthesis. It's going to open sodium and calcium channels, and it's the sodium that comes in and generates the impulse. Okay. Calcium causes decreased response to a sustained stimulus, and that's adaptation. And that's why people can't smell an odor after being exposed to it for a while, right? So I always go the example of going to the vet, right? You take your dog to the vet, and the first thing you smell is urine, right? Right? Because, dogs get scared and they pee at the vet, right? Well, after about maybe half an hour, right, the urine smell is gone. Well, what happened? Did the smell really go away? No, it's because of adaptation, right? You become used to the smell, so you don't smell it anymore, right? You can see how this second receptor pathway works, right? You have the ligand binding the receptor. What happens now is the G protein bound to it kicks out the GDP, binds GTP. It becomes now motile along the membrane, is going to activate adenylate cyclase, and it's going to turn around and open calcium and sodium channels, right? And that results in a depolarization. Okay. And at threshold, hold, we generate an action potential. Okay. And remember that every step along this way is amplified, right? I talked about the fact that these kind of pathways are what are called cascades. Okay. And cascades have several steps, and at each step, you geometrically amplify the signal because this activator receptor can activate more than one G protein, which can activate more than one adenylate cyclase, which can make more than one cyclic A&P and open more than one ion channel, right? And so at every step, you get more and more and more of this, okay? And so you get this amplification, okay? And the advantage of amplification is that it grows quickly, okay? And also it is easy to stop, right? And the reason for that is that you can stop the first and or second path in the pathway. You quit manufacturing the result, which in this case is the opening of the channels. And there are other enzymes in here that will then defuse the cyclic A&P, for example. An example that's called phosphodiesterase. Okay, it tears apart the cyclic A&P, and then those channels close up, right? And that's the end of the signal. Okay, so you can see here again the odorant binding, the G protein sliding over, adenylate cyclase converting ATP to cyclic, and then we pop the channel, right? Now, the filaments of the olfactory nerves are going to hit the mitral cells. Those are going to synapse within the glomeruli of the olfactory bulb, and those mitral cells are going to project back along the olfactory tract to the temporal lobe of the brain, right? Amacrine cells release GABA to inhibit mitral cells so that only excitatory impulses get transmitted. So they're like the fine tuning. Okay. The activated mitrals are going to, again, project to the olfactory cortex in the temporal lobe. Some of this gets sent to the frontal lobe and some passes through the thalamus first so that the smell is consciously interpreted and identified. Okay. Some information goes to the hypothalamus, the amygdala, and the limbic system to elicit what are called emotional responses. Okay. Remember here, some passes through the thalamus. All right. That is primarily for the limbic input. Okay. But again, the bulk of it bypasses the thalamus and goes straight to the temporal lobe. We talked earlier about anosmia, which is the loss of the sense of smell that can damage the olfactory nerves. Examples of neurological disorders like Parkinson's can lead to this, also trauma to the olfactory nerve itself, okay? Sometimes we get smells where there are none, and these are sometimes caused by temporal lobe epilepsy in the olfactory cortex. And some people have olfactory auras prior to seizure, okay? And so what that is that's a sensation and a perception that's created at the level of the cortex and is piped to the frontal lobe of the brain. Okay, so that's a different kind of animal. But a lot of times in anosmia, when you damage the olfactory nerve, it grows back and you recover your sense of smell, okay, for reasons that we've already outlined, right? Now, taste is a little different, right? In taste, what happens is that we have structures called taste buds that are on the lateral margins of the papilla of the tongue, right? The papilla are those little things that stick up on the top of your tongue that are basically grippers for your food. Okay, you can feel them if you rub the top of your tongue against your upper lip. Okay, and they come in different shapes. There's fungiform, foliate, and vallate, and they're named for their shape, right? Fungiform are shaped like a toadstool. Okay, foliate are shaped like leaves, and vallate have a V formation on the back of the tongue. Okay, so that's what those are about. And then there are filiform papillae. They're shaped like filaments that don't have taste buds. Comma, but do provide texture and temperature information about your food. Okay. And then what cranial nerves deal with this? Okay. Well, 7, 10, and 9. Okay. Those are the 3. So that's going to be vagus. Okay. Glossopharyngeal and facial, right? It's facial. Facial. Come on. And glossopharyngeal. Okay. Those are the 3. Here's your tongue. And it's showing you, about where the papillae are found. Okay. Don't confuse the papillae with the taste buds. Okay. The papillae are the projections, right? And the taste buds are on the lateral margins of the papillae, right? Each taste bud has hundreds of epithelial cells. Some are gustatory cells, which are the taste receptors that also have microvilli known as gustatory hairs projecting into taste pores covered in saliva. These are sensory dendrites that are coiled around. There are sensory dendrites coiled around the gustatory cells, and they send taste signals to the brain. Okay. The 3 types of gustatory cells, one releases serotonin, others don't have synaptic vesicles, but one releases ATP as a neurotransmitter. And then there are basal epithelial cells that are also stem cells. Okay. So setup is similar to olfactory epithelium. Okay. But in this case, the gustatory cells are receptor cells, not neurons. Okay. That's the only real difference. You can see where they are on the margins of a taste bud, right? And then if we look deep down, you can see the different cell types, right? These are the cranial nerves that are projecting into the taste bud, all right? There's your taste pore right there. That's how the taste that dissolves in the saliva gets in. The gustatory epithelial cells, the ones that do the work, are these dark guys, okay? Notice that they're all associated with the neurofilaments, right? So they can send their information back ultimately to the brain, right? And then you've got these supporting cells, okay? And those are the, you got the epithelial cells that regenerate. These are stem cells here, okay? And then you've got supporting cells that help them out, okay? What are the tastes? Sweet, sour, salty, bitter, and then something called umami, which is meaty, okay? How do they work? Well, they're going to work primarily through second messengers for all tastes except sour and salty, which directly open ion channels. Okay. In case of sour, protons are the acting ion. In case of salty, it's cations. Could be potassium, could be sodium, okay, etc. Okay. So there might be a sixth taste, which are long chain fatty acids from lipids. Okay, we don't really know yet. These tastes guide our diet choices for beneficial and harmful substances. A dislike for sourness and bitterness is a protective way of warning us if something is spoiled or poisonous. Okay, so you have to think back in the day, right? Early in evolution, how did we shop? Well, we just went out and ate stuff, right? And some stuff tasted bad. Some stuff tasted good. Some stuff had no flavor. Okay. But all of these, were among the population, and what was bad to one person might be good to another, right? But what happened over time was that the people that thought bitter and sour were in the good category, right? And that sweet and umami were in the bad category or had no flavor, right? Right. What happened to those individuals? Well, these individuals died, right? And did not contribute their genes to the subsequent population. They were wiped out, okay? Which makes perfect sense, right? Natural selection. And then those individuals that enjoyed the taste of sweet, umami, salty, what happened? They survived. And then they passed those genes on to their offspring. And so that's why we like what we like, okay? We like high calorie targets, okay? And the reason is, again, back in the day, the way that our eating pattern, right, was very different than it is now. Now, if we want food, we go to the grocery store and we buy it, right? Very simple. But back then, we were what are called hunter-gatherers. And so the way that we shopped was that we sat in the cave or in the field and we watched. And then an object would come across our path, like a yak or an elephant or a cow or something else that we might be able to kill and eat, right? And then we would run out and we would kill it and eat it, okay? And the individuals that enjoyed the flavor would get the lion's share of the food, and they would live long enough to reproduce because they'd be fed, because they'd also live long enough to get to their next meal, right? Those that didn't enjoy the flavor, guess what? They didn't eat, they died. They didn't reproduce, okay? So it's natural selection that makes us like high calorie targets, okay? Nothing more than that. So this is why we eat what we eat. And then what happened was we invented something that was unexpected and happened a little bit too fast for evolution to adapt to it. We invented farming. Okay. What is farming? Farming is going out in the wild and picking out animals and plants that you like the taste of or like the products of, gathering the seed in the case of the plants, or breeding the livestock in the case of the animals, and producing lots and lots of food that you can eat, meaning eventually you had a food surplus, right? Farming led to food surplus, right? Which led to what? Trade. Which led to what? Civilization. Okay. Sort of a straight line there, right? So here now we jumped way ahead in terms of what our diet is, and the process of evolution hasn't kept up. And so we still love these high calorie targets, even though there is an abundance of them. And so this is one of the things that leads in the modern day to obesity. Okay. Obesity. Okay. So you're fighting evolution. Okay. So don't feel too bad if it's tough to keep the weight off, right? Because back then, you know, it was eating in a very random manner and doing lots of exercise to get your food, right? Preparing it, killing it, all that stuff. Okay. So they tended to be in shape, right? To be able to taste a chemical, it has to dissolve in saliva, diffuse into the taste pore, and get in contact with the gustatory cells. Okay. The taste, it depolarizes the cell membrane and causes neurotransmitter release that binds to the afferent fibers, neurofilaments, and then generates an action potential that goes back to the brain via the thalamus. Different gustatory cells have different thresholds of activation. Bitter are the most sensitive, and that's nice because that warns us of dangerous stuff, right? We all adapt in around 3 to 5 seconds, with complete adaptation in 1 to 5 minutes, right? And so I talked about what it is that generates the transduction, right? Sodium for salty, protons for sour, right? And then all these others, sweet, bitter, umami, they're all G protein activated, okay? And the G protein is called gusducin, all right? This causes stored calcium release. This opens cation channels. This lets in sodium, okay, or protons, all right? And that results in a depolarization, and that can generate neurotransmitter release, okay? And those are the 3 nerves, right? They've got the nerves messed up. Vagus nerve is 10, okay? And glossopharyngeal is 9. There you go. Let's see here. No, glossopharyngeal 7. Facial 7, glossopharyngeal. Yeah, that's 9. Yep. Fiber synapse in the solitary nucleus in the medulla oblongata in the brainstem, travel to the thalamus, and then to the gustatory cortex in the insula, the 5th lobe of the brain. The hypothalamus and limbic system are involved and allow us to determine our appreciation of the taste, right? So there's the pathway, okay? So those different nerves project back to the medulla. Another set of nerves is going to project from the solitary nucleus back to the thalamus. And then the third set is going to project to the gustatory cortex in the insula of the brain, right? And then what will happen is that, just like all the other general and special senses, is that we will get first sensation, which is the idea that something's happening, and then we will draw on memory to attach perception to it, right? So there's going to be an association area in the insula that we're going to draw on as well, okay? Important roles of taste: trigger impulses in digestion, like increased secretion of saliva into the mouth, increased secretion of gastric juice into the stomach, and can initiate gagging and vomiting. Okay. Taste is 80% smell in humans. Okay. In humans. Okay. So if our nose is blocked, food tastes bland because we're not getting the olfactory input. Okay. Such as when you have a cold or sinusitis. Okay. The mouth has thermoreceptors, mechanoreceptors, nociceptors in it as well for other information like temperature and texture that can add to the experience of taste. Spicy hot foods hit pain receptors in the mouth, which some people like. I don't. Okay. The active ingredient, for instance, in chili peppers is called capsaicin oil. Okay. And it is non-polar. Okay. So it can go right through cell membranes of nociceptors. Okay. And that can generate what? Pain for many people. Some people dig it. I used to have a boss that would eat raw habanero peppers and not even blink, right? And that has to do as much with the composition of the taste bud proteins that's genetically encoded for, as it does the way that the brain experiences the different flavors and the associations the brain makes with it. Okay? That's why some people like some foods and some people hate that very same food. Okay. Taste disorders are less common than smell because taste receptors are served by 3 different nerves. It's not likely that all 3 would be damaged at the same time. Causes of taste disorders include upper respiratory tract infections, the reasons that we just indicated, you lose the olfactory part, head injuries, chemicals or meds, okay, and head and neck radiation from cancer treatment. And we should also add age. What happens with age? Taste buds lose function. Why is that? Because the death rate is greater than the regeneration rate for olfactory neurons and gustatory cells. So you lose input. It's one of the reasons sometimes older people overseason their food so they can taste something, right? So one of the problems this leads to is a bland flavor in most food, which means that you are less likely to eat. And unfortunately, that means less energy production. And what does that lead to eventually? Death. Yeah, it's all related. Don't mean to be a downer, but this is what happens. Okay. That's a lot of times why you grow into some flavors, right? Your taste buds are and your olfactory epithelia are much more sensitive when you're young than when you start to age, right? And so some stuff that used to taste awful tastes much different when you get older, right? So let's take 10 and we'll come back for the second half of this and we'll talk about the ear, okay? So, Okay, we're back. All right. So now it's on to the inner ear. Okay. We want to go with a share. Let's just do desktop for now. Share. All right. So we've talked about hearing and balance a little bit in lab already. And we talked about the fact that, remember from the model in lab, that there are three parts to the ear. There is an inner, a middle, and an outer ear, right? The boundary between the outer and the middle ear is the tympanic membrane or eardrum, okay? And the boundary between the middle ear and the inner ear is the oval window, which is on the surface of the cochlea, okay? The middle ear and the outer ear are designed for hearing. Well, the inner ear is hearing and equilibrium. And you might say, well, what's equilibrium? And that's our sense of balance. So equilibrium is balance. Okay. So if we look at the outer ear, the auricle is like the funnel. It's the part of the ear that you can see on the head. The helix is the rim. That's the upper part of the ear, and the lobule is your earlobe, where your earrings go, okay? The external acoustic meatus goes through the temporal bone and is the tunnel that leads to the tympanic membrane, which is the eardrum, okay? It's lined with sebaceous glands and hairs, okay? And ceruminous glands, all right, which make earwax. The purpose of the hairs and the sebaceous glands is, again, to trap particulates. Okay. The purpose of the earwax is to trap particulates retard mold growth, and repel insects due to the bitter taste. Right? The meatus also transmits sound waves to the eardrum. Okay? So the tympanic membrane is the boundary between the external and the middle ear. It's connective tissue, vibrating in response to sounds and transmitting the sound waves to the ossicles of the middle ear. Okay, there's three ossicles, right? You can see them here. They are the malleus, sometimes called the hammer. Okay, the incus, which is sometimes called the anvil, and the stapes, which is sometimes called the stirrup. Okay, these are all bones, right? And they are designed to transmit and concentrate the sound to this little area here on the cochlea, which is a thin membrane called the oval window. Okay. The cranial nerve that deals with all this is cranial nerve 8. Which is the vestibulocochlear nerve. And in some older textbooks, it's called the auditory nerve. Okay. So you can see the different structures. Okay. This, of course, is the cochlea. Okay. These are the semicircular canals. And then this little sort of enlarged area here, this is called the vestibule. Okay. The middle ear is an air-filled mucosal lined cavity in the temporal bone with the eardrum on the lateral side and the oval and round windows on the medial side. There is a recess, the epitympanic recess in the upper portion of the middle ear, which is the roof of the cavity, and a mastoid antrum, which is a canal for communication with mastoid air cells in the mastoid process. What's the mastoid process? That is the bump behind your pinna, your external ear. Okay. You can see the ossicles here, right? The eardrum, the location of the oval window. Okay. And then remember, this is a branch of cranial nerve 8. Okay. These are both branches of cranial nerve 8. And then the auditory or eustachian tube connects the throat to the middle ear. So that's to regulate pressure on either side of the drum so that it can vibrate. Okay. The ossicles transmit the sound waves to the oval window, okay, and concentrate the power of those sound waves, okay. There are a couple of muscles in the region, the tensor tympani muscle and the stapedius muscles will contract in response to loud sounds in order to prevent blowing the ossicles apart or ripping the eardrum, okay. There's also ligaments that connect those muscles to their attachment points, okay. And there are synovial joints between each of the ossicles, right? You can see here, again, a close-up of the medial to lateral view of the middle ear, okay? And you can see the attachment points for the tensor tympani and the stapedius muscles, okay? Again, this is all embedded in the temporal bone. Okay. We talked about otitis media. What happens in otitis media is you get an infection in the middle ear that often comes via the eustachian tube. The bacteria then grow behind the eardrum, creating gas, which pushes out on the drum and can eventually rupture it. Okay. At that point, the pain caused by otitis media goes away, but the eardrum will heal with scarring. Right. So one of the things that you can do is to treat this long term is to use what are called near ontogamy tubes. Okay, which you probably know better as tubes in the ears. Okay, and the purpose is to equalize pressure. Around the eardrum. And also discourages growth of bacteria. Okay. The tubes generally are ejected once their surgical opening heals on the lateral side of the drum where they can be rescued. Okay. These days, they primarily treat with antibiotics, but in the old days, they would lance the drum and you would use Aralgin for pain. Okay. There's a labyrinth in which the internal ear structures reside. It is basically a series of openings connected within the temporal bone behind the eye socket. The bony labyrinth are channels and cavities that work its way through the temporal bone, divided into the vestibule with semicircular canals and cochlea. The cochlea is filled with perilymph externally and endolymph internally, and the remaining structures, the vestibule and semicircular canals, are filled with endolymph. Okay. The membranous labyrinth is a series of membranous sacs that are contained in the bony labyrinth and are filled with the endolymph, which is unusual in that it is potassium rich. Okay. So you can see the structures here. All right. What are the sensory inputs that make these organs work? Well, for the cochlea, it is sound. Okay. For the vestibule, it's gravity. And for the semicircular canals, it is movement, specifically head movement. Okay. The vestibule is for static equilibrium. Okay. It has a bony labyrinth and a membranous labyrinth that contain the saccule and the utricle. Okay. The utricle is horizontal in anatomical position, and the saccule is vertical. The sacs have equilibrium receptors known as maculae that respond to gravity and the position of the head, as opposed to the movement of the head. Okay. The semicircular canals are oriented in the three planes of the body, and have swellings called ampullae at their bases, which house special sense organs called the cristae ampullaris. And these are hair cells that have their apical margins embedded in something called the cupula. Okay. And they respond to rotational movements of the head as fluid bends the cupula and bends the hair cells. Okay. When the hair cells bend, ion gates open. Right. In the vestibule, gravity pulls on the otolithic membrane that is covering the apical surface of the hair cells in the maculae, and that opens ion channels, and that's what produces a action potential. Okay, so you can see those structures here, right? Those are the sensory organs or the semicirculars. You can see them there. Okay, at the bases of each of these things, and there are the utricle and the saccule. Okay, there is the utricle. Okay, and there is the saccule. Okay, and those are going to respond to gravity. Okay, while these respond to head movement. Okay, and of course, again, these respond to sound. Okay. The cochlea is a bony snail-shaped chamber extending from the vestibule, coiling around a pillar called the modulus, containing an interior cochlear duct, inside of which is the organ of Corti, which actually does the transduction. That's where the hair cells live. And then there's going to be a scala vestibuli and a tympani peripheral to the cochlear duct, which contain the perilymph. Okay. So you've got perilymph on either side, you've got endolymph in contact with the hair cells, and this is high in potassium. Okay. The scala tympani and the vestibuli are continuous at the helicocentrema, which is the very tip of the cochlea. So the fluid-filled cavities of the tympani and the vestibuli are connected to each other, right? The vestibular membrane is the roof of the cochlear duct that separates the media from the vestibuli, right? And the basilar membrane is going to be the floor of the cochlear duct, okay? The stria vascularis is the external wall of the cochlear duct made up of mucosa, which makes the endolymph. The floor of the cochlear duct, as we said, is the basilar membrane supporting the organ of Corti. Okay. The spiral organ has cochlear hair cells that are arranged in one row of inner and 3 rows of outer hair cells. The hair cells are between the tectoral and the basilar membrane. The tectoral membrane is on the top. The basilar membrane is on the bottom. Okay. The basilar membrane vibrates, okay, in response to certain sound frequencies. The cochlear branch of the vestibulocochlear nerve runs from the organ of Corti to the thalamus, the medial geniculate nucleus, and then from there to the temporal lobe of the brain, where the auditory cortex is located. All right, so you can see here a cut through the cochlea, showing you the different structures, okay, how they're arranged. All right. And then you can see here the 3 chambers, right? There's the cochlear duct, right? There's the scala vestibuli, and there is the scala tympani. Okay, perilymph in these, okay, endolymph in the cochlear duct. Okay, where is the basilar membrane? It's right there. Where's tectoral membrane? It's up here. Okay, where are the hair cells? Between. Okay, you can see a close-up here showing you the hair cells. Note they are associated with afferent nerve fibers, right? So what's going to happen is that sound waves are going to cause vibration of the basilar membrane. These guys are going to vibrate up and down, right? And when they do, these hair cells are going to bend, right? And when these hair cells bend, that's going to open ion gates and potassium is going to flow in, right? And that's going to ultimately result in neurotransmitter release, and that will generate an action potential that's going to go back to the thalamus of the brain, okay, of the diencephalon, let's be more clear, right? And that's what it looks like in the EM micrograph. Pretty cool, okay? These hairs can die, and then you go deaf, right? That stinks. And that's when you have to get something called a cochlear implant. A cochlear implant attaches to cranial nerve 8, which attaches to thalamus, which attaches to the temporal lobe. Okay, so it bypasses the cochlea, becomes your new cochlea, right? So those are the structures, right, and what they respond to. So let's take 10 more minutes, and then we'll come back to the first part of part D. Okay. All right. So let's share desktop here. Begin here. Share. There you go. All right. So now we're going to get into the guts of what goes on in here in the balance. Okay. So the idea with hearing is that we're going to take a pressure wave, which propagates through gas by compressing particles, right, in a wave-like fashion. So you have a region of compressed and then uncompressed particles, and that wave propagates through gas, all right? And that's what sound basically is, okay? It also can propagate through water, any liquid or gaseous medium. And that's going to eventually hit the tympanic membrane, okay, and cause it to vibrate, right? Those waves of pressure difference, okay? So what'll happen is that the area of high and low pressure will hit the tympanic membrane and cause the membrane to vibrate, okay? So the object vibrating causes waves to move outward in all directions. The air around it is a compression wave that propagates through the medium, and then it eventually will decline with time and distance, okay? But eventually, what'll happen is that that pressure wave is going to reach the tympanic membrane, right? So you can see, for instance, with a tuning fork, right? It's moving, the sound waves are being created as gas molecules are being compressed, and then in between are areas of low pressure. So you got high pressure, low pressure, high pressure, and that is the characteristic of the wave here. Okay, the frequency of the wave is the number of these high pressure areas that pass a particular point in 1 second, and it's measured in hertz, which is Hz. Okay, so frequency is indicated as the Greek letter nu. Okay, and it's measured in hertz. Okay, and the wavelength is just the difference from peak to peak. Okay, wavelength, and it's measured in, depending on what kind of wave you're talking about. Often it's measured in nanometers. Okay, and sometimes it's measured in micrometers. Okay, all depends on the scale of wave you're talking about, and the relationship between frequency and wavelength is always this, right? The inverse of the wavelength is equal to the frequency. Okay? So you can see radiation of a sound wave heading out, right? We talked about the frequency and the wavelength, right? The frequency is going to determine the pitch. Okay. All right. And the wavelength is also going to be related to the pitch. Okay. But the amplitude of the wave is going to be the loudness, the power of the wave, and that's measured in something called decibels. Okay. Frequency range for human is between 20 and 20,000 hertz, but it's most sensitive between 1,500 and 4,000. We talked about the pitch. Okay. The quality is the mixture of sounds, mixture of different frequencies and different volumes. Okay. A tone is one frequency, whereas a sound is frequently many frequencies and many different amplitudes. Okay. So you can see here some examples of a complex sound, right? Low frequency, high frequency. Okay. The high frequency wave here is the red one. The low frequency is the blue one. Okay. And that's your pitch. Pitch. Okay. The amplitude is the height of the crests and is measured in decibels, right? The higher the amplitude, the greater the volume. Okay. Normal range between 0 and 120. Normal conversion around 50. Normal conversation around 50. Pain is about 120. Okay. So people that work at airports, jet engines frequently exceed that. That's why they wear personal protective equipment, which are headphones around the ears, noise canceling headphones. Okay. You can have hearing loss when you're exposed to high amplitude sound waves. You can knock those hairs right off the cells of the basilar membrane of the cochlea, right? And you lose that region of your hearing, right? So you can see here, again, amplitude and intensity, right? Loud and soft is a difference in the peak of the wave, okay? The tympanic membrane is what catches it, right? The higher the frequency, the more vibration you have, the ossicles are going to transfer that to the oval window, okay? The tympanic membrane is 20 times larger than the oval window, so this amplifies the sound 20 times, and that gives it enough kick to move the fluid, the perilymph, inside the cochlea, right? The scala vestibuli is going to be in contact with the stapes that rock back and forth on the oval window in each vibration, and that causes wave motion in the perilymph. The wave ends at the round window that relieves the pressure. The helicocentrema are the waves with frequencies below the threshold of hearing that travel through it and the scala tympani to the round window. So they don't even shake the basilar membrane. But the basilar membrane will vibrate, okay, in response to what we call resonant frequency. Okay. What is resonant frequency? Well, every object has a built-in resonant frequency, which is the frequency of wave that basically will blow the object apart. You may have seen opera singers hit those high Cs and shatter the wine glass. Used to be a Memorex commercial for Memorex cassette tapes, okay, back in the olden days. Well, the resonant frequency of the basilar membrane varies from the part that's closest to the drum versus the part that's closest to the helicocentrema. Okay. So the part that's closest to the drum, that is for high frequency, and helicocentrema is for low frequency. Okay. So the membrane properties, the resonant frequency of the basilar membrane changes as you go from the drum end to the helicocentrema end. Right? So you can see here that you get a particular pitch that's going to wiggle the basilar membrane in that region only, right? And sounds of different frequencies are going to wiggle the basilar membrane in other regions, okay? So you can see here how the sound wave is propagating through the perilymph, which is going to wiggle the endolymph, which is going to wiggle the basilar membrane, and that's going to cause the hair cells to bob up and down, and they're going to hit their heads on the tectoral membrane, which is going to be essentially on the top of those hairs, and that's what's going to cause those hairs to bend and open the ion gates, right? So you can see here, sound waves hit the ossicles, pressure waves go through the scala vestibuli in the perilymph, sound with frequency below the hearing range just zip right around in the perilymph and come back the other way. But in the hearing range, they go through the cochlear duct and vibrate the basilar membrane at the resonant frequency of the sound and the resonant frequency of the membrane that corresponds to it. Okay. That's how the sound, quote unquote, knows where to go. Okay. The basilar membrane changes along its length. The fibers near the oval window are short and stiff and are for high frequency, and near the apex, they are longer and floppier, and they are for low frequency. Okay. You can see if we unroll the membrane, what I was talking about, right? High frequency near this side, low frequency over here. Okay. And then you can see how the fibers change and the resonant frequency changes, right? Here, the resonant frequency is high, and here the resonant frequency is low. Okay? Inner hair cells move when the basilar membrane bobs up and down in response to the endolymph bobbing up and down. Okay. The stereocilia are embedded in the tectoral membrane. They're connected to each other by what are called tip links. It's sort of like a rope between the tops of the stereocilia. When the tip links are then pulled on, the ion channels open and the potassium rolls in. And that's what's going to make our action potential, right? So you can see right there, there's our hairs, right? That's attached to the ion gates. And that is what's going to bend and open them up. And then in comes our potassium, right? Our potassium is out here because this is high potassium concentration, okay? Potassium concentration is what that means, okay? It's going to go right into those cells and result in depolarization, okay? So the bending of the stereocilia is going to pull on the tip links. Potassium and calcium is going to open. They're going to flow into the cell. They're going to generate that receptor potential, and that can lead to neurotransmitter kickout. In this case, the neurotransmitter is glutamate, okay? If there are enough glutamate released, action potential happens. Afferent neuron, cochlear branch of cranial nerve 8, carries that back to medial geniculate nucleus of the thalamus, okay? Then the ion channels close, and we start all over again, right? So you can see how this is working, right? There's the tectoral membrane up top. This is the vibration down here in the basilar membrane, right? And then when we open these ion channels, right, in flows this stuff, and then we get neurotransmitter release down here, okay? And then when we go back to not bending the stereocilia, we close the ion gates and no more neurotransmitter is released, right? Until the next wave comes along, okay? The outer hair cells are efferent neurons that convey messages from the brain to the ear. The outer hair cells can contract and stretch and change the stiffness of the basilar membrane, which fine-tunes the inner hair cells by amplifying the motion of the basilar membrane and protects inner hair cells from loud noises by decreasing the motion of the basilar membrane, okay? So the outer hair cells protect and tune, right? The inner hair cells transduce sound. Okay? Okay, how do we get this back to the brain? Well, the pathway, right? Spiral ganglion, which are in the cochlea, embedded in the basilar membrane. Project to the cochlear nuclei in the medulla, which project to the superior olivary nuclei at the pons medulla junction, which hit the medial lemniscus, which is a white matter tract. White matter ascending tract in the brainstem, right? Which hits the inferior colliculus, which is in the midbrain, okay? Which projects to the medial geniculate nucleus of the thalamus, okay? Which projects back to the primary auditory cortex, which is where? The temporal lobe of the brain. Okay, that's the pathway, right? Some of the fibers cross, some don't. So both auditory cortexes get input from both ears, and that's your pathway, sort of diagrammatically, right? This is cochlea, here, right? Here we go back, spiral ganglion, right? And then we're going to hit bipolar cells, and then cochlear nuclei, then olivary, and then up the lateral lemniscus. There's your thalamus, right? And then there is your cortex, which is temporal lobe. Okay. And that's going to provide sensation. And then with the temporal or the auditory association area, we get perception. Okay. The perception of pitch is the result of impulses from hair cells in different positions along the basilar membrane, interpreted by the brain as different pitch. And loudness is determined by the brain as it increases in the number of action potentials that result when the hair cells have larger deflections. Okay. In other words, are open for a longer time, resulting in more depolarizations per second, which means a stronger input. Okay. The localization of sound is going to depend on the intensity and the timing of the sound waves. The timing is increased on one side, the brain interprets the sound as coming from that side, right? The next big role of the inner ear is equilibrium, which is our balance, right? The vestibular apparatus, which is in between the semicircular canals and the cochlea, is going to respond to static equilibrium, which responds to gravity. Okay. Well, the semicircular canals are dynamic equilibrium, which respond to movement of the head. Okay. You can see the structures here. Okay. Nothing new, except this time we're concerned with what goes through this branch of the vestibulocochlear nerve, the vestibular nerve, which gets information both from the semicircular canals and the vestibule. Okay. So when we come back in 10 minutes, we will finish this out. Okay. So let's do that. Okay. The maculae are in the vestibule, and they contain hair cells that are embedded in something called the otolithic membrane, which is a gel-like substance that has crystals of calcium carbonate in it to give it more weight, so that it responds more easily to gravity. The other thing you have to remember is that the vestibule and the semicircular canals, along with the cochlear duct, are all filled with endolymph. Okay. The maculae respond to gravity. They respond to the position of your head in space, and they also respond to what we call linear acceleration, but not rotation. Okay. You can see here again, drawing where are the maculae. Again, the maculae are down here. They are here, here, and here. Okay. Okay. Again, gravity. Okay. Each macula is epithelia with hair cells that contain stereocilia and kinocilia on the apical end, the top. Okay. The stereocilia are in the otolithic membrane, which contains those calcium carbonate stones for extra mass and allow it to respond more effectively to gravity and changes in gravity, right? The utricle maculae are horizontal. They have vertical hairs and they respond to changes in the horizontal plane. The saccule is vertical and responds to up and down movement. Okay. The hair cell synapse with the vestibular nerve, which is the vestibular branch of the eighth cranial nerve, located in the superior and inferior vestibular ganglia, okay? And you can see here what the otolithic membrane looks like in the utricle saccule, and you can see where the hair cells are located and where the afferent nerve fibers are, okay? So this is all still eighth cranial nerve, right? The hair cells release neurotransmitter all the time, but when you have a change in linear acceleration, we change the action potential frequency to the brain. The density of the otolith membrane makes it lag behind the movement of the hair cells when the head changes position. The base of the stereocilia moves at the same rate as the head, but the tips in the membrane are pulled by the lagging membrane and the hair bends. The main point being the hair bends, okay? Ion channels open, potassium flows in, calcium flows in, okay? And we have depolarization. The bending of the hair cells in the direction of the kinocilia depolarizes that cell, increases neurotransmitter release, more impulses travel up that vestibular nerve to the brain, while bending away has the reverse effect, okay? Reducing the rate of impulse generation, and you get less of a stimulus perception, okay? So you can see here how we bend the head and the cilia respond accordingly, okay? Difference in action potential, okay? So you can see how this would be different bending your head forward and back versus bending your head to the left and the right, okay? All of those are going to be recorded in the maculae that are in the vestibule, okay? Now, the cristae ampullaris is different, okay? The cristae ampullaris are hair cells that are embedded in something called the cupula, which bends in accordance to the movement of endolymph past the cupula that bends those hair cells embedded in the cupula, okay? So this is rotational movement of the head, all right? The cristae changes in velocity to rotational movement. The inertia in the cupula causes the endolymph in the semicircular canals to move in the direction opposite of the body's rotation. The hair cells bend, okay? And you can see here what the cupula looks like. Remember I told you it looks like a cupcake, right? This is the icing of the cupcake, the cupula. This is what's going to bend in response to the fluid moving past it, right? And this is like the cake part of the cupcake, the nerve fibers, okay? And this would be the top of the cupcake, okay? All it's one sensory organ, okay? And there's what it looks like in the micrograph, okay? That's a crista ampullaris, and they are in the ampulla, which are the swollen bases of each of the semicircular canals, okay? The bending of the hairs in the crista ampullaris makes the depolarization. Rapid impulses are going to come to the brain quicker, and bending of the hairs in the opposite direction, hyperpolarization, fewer impulses. So the brain knows about the rotation of the head. So the brain's getting information about balance either while you're moving or while you're standing still, okay? The axes of the hair cells in the complementary semicircular canals are opposite. So when depolarization occurs in one ear, there's hyperpolarization in the other. The endolymph comes to rest after a time, so it detects only changes in movement. So you can see some examples of what happens, for instance, when we do an axle, okay, on the ice rink. You're going to get a different response on one side versus the other, okay? Nystagmus is where impulses from the semicirculars are linked to reflex movement of the eyes that keep tracking even though you're no longer moving. It's an eye movement during and immediately after rotation that can be accompanied by vertigo, right? As rotation begins, the eyes drift in the direction opposite the rotation because it's trying to track what's in front of you, okay? And the central nervous system compensation causes a rapid jump towards the direction of rotation. As the rotation ends, the eyes continue in the direction of the spin, then jerk in the opposite direction, okay? Equilibrium information goes from reflex centers in the brain stem, allowing fast responses to imbalance so we don't fall down. Impulses from activated vestibular receptors travel to either vestibular nuclei in the brainstem or to cerebellum. The three modes of input for balance and orientation are the vestibular, the visual, and the somatic receptors, right? And all this is going to go to cerebellum so that we don't have discoordinated movement, right? Somatic receptors are what? These are things like Golgi tendon organ, and muscle spindle. Okay. Visual receptors, obviously photoreceptors. Okay. And vestibular receptors, we're talking about hair cells. Okay. So you can see how this is working, right? We take all of this, okay, these three different pieces of information, we pipe them over to the cerebellum, right, which is going to compare, right, what you're doing with what you think you're doing, okay? Compare actual and intended movement, okay? Vestibular nuclei are going to project to the nerves that project to the muscles that move the eyeball, and then spinal control to the neck, limb, trunk, and muscles that control the orientation of your gaze, okay, as well as your overall balance, right? And remember that the cerebellum is also going to connect to what? The cortex, motor cortex, right? And that's how it knows to correct actual versus intended, okay? Equilibrium problems are unpleasant, causing nausea, dizziness, loss of balance, nystagmus in the absence of rotational stimuli happens. I experience that myself. Motion sickness, which I also suffer from, is where the visual input and the equilibrium input differ. The information causes nausea. We see salivation, pallor, deep breathing, sweating, headache. We can treat with scopolamine. I've used that and it works, okay? And that's one of the drugs that they use for chemotherapy patients to suppress nausea, okay? Anti-motion drugs are going to depress the vestibular input, okay? Meclizine, scopolamine, so on, okay? All right, conduction and nerve deafness, the difference between the two, okay? Conduction deafness is a problem from the pinna all the way through to the ossicles, okay? Where sensorineural deafness is from the cochlea back to the brain, all right? So sensorineural deafness is essentially something that you can overcome with a cochlear implant, right? So we put this in the temporal bone, usually in the mastoid process. Okay. And we bypass the cochlea. Okay. And you relearn how to hear. Okay. So this is like your new eardrum. Okay. Plus inner ear. Goes right into the nerve, which of course projects back to the brain. Right. Tinnitus is the ringing sound that can be generated in the absence of any sound wave, and it can be the result of nerve degeneration in the cochlea, inflammation of the middle or internal ear, or it could be side effects of aspirin. Okay, tinnitus. Meniere syndrome is a terrible condition. It results in vertigo. Okay, and it's because what happens here is that the movement of the fluid in the labyrinth is impinged. Okay, you get vertigo, nausea, vomiting, nystagmus. Okay, we use motion sickness drugs or surgical removal of the labyrinth in some cases. And you can imagine what that leads to, okay? Taste and smell are functional at birth. Few problems usually occur until the fourth decade when these senses begin to decline. They get poor after age 65. And we talked about why that is, right? The rate of cell death increased rate of cell death compared to decreased rate of cell regeneration. So you lose function, right? Lose function. Okay. What else happens in vision, right? Optic vesicles, when we're forming the visual apparatus, protrude from the diencephalon, generating optic cups. The stalks forming the optic nerve. The lens formed from ectoderm, one of the three germ layers, okay? Vision's not fully functional at birth. Babies are often hyperoptic because the eyes are shortened. So they see only gray tones and their eye movements are uncoordinated. They don't generate tears for the first 2 weeks. By 5 months of age, they can follow objects, but the visual acuity is poor. Depth perception and color vision develop by age 3. Adult eye size is reached around 8 to 9 years of age, and around age 40, the lenses lose elasticity, and we see presbyopia, right? I've talked about that. That's the need for reading glasses due to stiff lens. So you can't focus on things near, right? With age, the lens can lose clarity, so we can have cataract. Dilator muscles aren't as strong. Visual acuity drops. Lacrimal glands get less active. The eyes can get dry and prone to infection, right? When we look at the sense of hearing, okay, it develops generally beginning in the 3-week embryo. The inner ears develop from ectoderm as optic or otic placodes, otic meaning of the ear, and then an otic pit and a vesicle form. The otic vesicle becomes the membranous labyrinth, and the surrounding mesenchyme is going to be the bony labyrinth, because we're going to find out later on that mesenchyme is derived from mesoderm, okay, which forms the musculoskeletal system, among other systems, okay? The middle ear structures develop from pharyngeal pouches, ossicles from neural crest, and pharyngeal cleft develops into outer ear structures. You might know, what the heck is neural crest? Well, neural crest cells, at some point, when you're still an embryonic disc, right, and you've got this little primitive streak that eventually is going to form a notochord, and then you're going to get, if we look at the disc sort of transversely, right, you're going to get this little groove with the notochord under it, right? The neural crest cells are here, okay? Okay, and just to orient you, that the amnion would be above this disc and the yolk sac would be below it. Okay, and this is what? This is you. Startling, I know, but there you are, okay? And the neural crest cells, what they do before the this groove here called the neural groove closes is that they sort of abandon ship, and then they fly all over the place, forming these special sense structures, as well as some structures in the heart and some structures in the craniofacial area, okay, among other places, right? By month 4, infants can turn their head towards the voices of family members. They have reflexive auditory responses. Language skills are usually tied to the ability to hear well. There are a few ear problems until your 60s when the spiral organ becomes less functional, right? And why is that? Same reason for all the other special senses. The rate of cell death outstrips the rate of cell regeneration. Presbycusis, which is old ears, you lose high-pitched perception first, and that begins sensorineural deafness, okay? High-pitched sounds have more energy than low-pitched sounds, and that's why those particular hair cells get the brunt of the deterioration due to force, right? Congenital problems of eyes are relatively uncommon, but the incidence increases with maternal infection. German measles can be damaging to the special senses, especially during the first 3 months of pregnancy. And there are problems associated with rubella, such as blindness and cataracts. Congenital abnormalities are common in the external ear. You might have missing pinna or absent external acoustic meatuses, so you got no pathway back to the eardrum. Maternal rubella can also cause sensorineural deafness, okay? It's thought maybe that's what affected Helen Keller, okay? So I have a set of videos. I'm going to run those for you tomorrow night in addition to our discussion on endocrine, okay? And of course, I'll post this. It's going to process after class is over. I'll post this and send you both the PDFs and the podcast, okay? And we'll go through taste and smell and vestibular and hearing one more time, just to make sure you've got it all. And then we'll concentrate primarily on endocrine, okay? Does anybody have any questions before we bump out? We're good? I will see everybody tomorrow night then, okay? Thanks for coming.